I'm from Autonomous University of Barcelona. Thank you. Good morning, everyone. The title of my topic today is Generation of Pigdendritic Cells from Flt3 ligand-dependent bone marrow cultures and the infection by PRRS1. Dendritic cell is very important in the immune system. They are the most potent cells in priming naive T cells, like a bridge between innate and adaptive immune response. Right now, 5 types of dendritic cells have been identified. Type 1 conventional, type 2 conventional, and plasmacytoid dendritic cells. These 3 types are from the same origin. Monocyte-derived dendritic cells and Langerhans cells are from the other regions. However, dendritic cells is a very rare cell population, less than 1% in all the tissues. That means it will be very difficult to get a good yield from in vivo isolation. For that reason, most of studies rely on the in vitro production. In pig studies, the most commonly used stimulus are GM-CSF and IL-4. However, with these stimulus, most of the cells generated are more like migratory dendritic cells rather than steady-state or bona fide dendritic cells. Then, how to produce bona fide dendritic cells? In my study, I used Flt3 ligand because bona fide dendritic cells are conserved with the expression of Flt3. And in human and mice, bona fide dendritic cells have been successfully developed by this Flt3 ligand culturing model. Moreover, a study in 2010 indicated that Flt3 ligand has the potential to produce pig bona fide dendritic cells. So the objective of my study is to derive and characterize pig bona fide dendritic cells from Flt3 ligand-dependent bone marrow culture system and assess their susceptibility to PRRS1 infection. For the production, I isolated bone marrow hematopoietic cells from the 4-week-old pigs and then culturing them with Flt3 ligand at 20 nanogram per milliliter for 14 days. During this time, half of the medium was changed every 3 days. The resulting cultures, we call them Flt3 DC. These cells were then characterized in terms of phenotype and functional behavior. So what is the phenotype of this Flt3 DC? For the phenotyping, we used 4 antibodies to do the primary gating. The antibodies are CD14, cadm1, MHC2, and CD172A. After gating the singlets and excluding cell debris, a big population of cadm1 positive cells was observed. This population can be divided into two subsets based on CD14 negative and CD14 positive. The further gating based on MHC2 and CD172A showed that within this CD14 negative gate, the majority of cells are MHC2 positive and CD172A positive. This is compatible with CDC2. And there is a small population is CD172A negative or low expressed. This is compatible with CDC1. By contrast, within this CD14 negative gate, most of the cells are MHC2 and CD172A double positive. Here we call them CD14 positive dendritic cells. To summarize, this Flt3 ligand culturing system produced 3 types of cells: a big CDC2-like population, a big CD14 positive population, and a minor CDC1-like population. In the following, we sorted these 3 populations and did further analysis. The further phenotyping, we used a panel of these 5 molecules. In this analysis, the data on CDC1 was lagged because the insufficient cell number. And by comparing CDC2 and CD14 positive dendritic cells, we see their difference is mainly on the expression of CD1, CD163, and DEC205. We see DC2 as CD1 positive. CD1 positive, CD163 negative, DEC205 low expressed, while CD14 positive DC as CD1 low expressed, CD163 positive, and DEC205 negative. We further checked the mRNA expression of some important dendritic cell markers. Flt3, the conserved dendritic cell marker here, is highly expressed on CDC1, lower on CDC2, and much lower on CD14 positive population. ZBTB46, which can be used to distinguish conventional dendritic cells from other immune lineage, here also highly expressed on CDC1, lower on CDC2, and much lower on CD14 positive DC. About the other genes, those 3 genes in the lower panel are highly expressed on CDC1. And this FC receptor gene almost exclusively expressed on CDC2. And all of these are consistent with what have identified on pig, human, or mice dendritic cells. But there is also one exception, IRF4, a transcription factor supposed to promote CDC2 development in mice. However, in our cells is highly expressed on CDC1. This might be due to the variation between different species. To optimize the production, we increased Flt3 ligand to 100 nanogram per milliliter. We added stem cell factor to the culture. However, no big effect on the phenotype, but with 20% increase of the yield when introducing stem cell factor. Moreover, based on the methods used on mice and human, we also tried a 2-step procedure with culturing bone marrow cells with Flt3 ligand and SCF to expand precursor cells firstly, and then introducing GM-CSF and IL-4 for further stimulation. However, with this method, no CADM1 positive population was observed. So in the following experiment, we stick on the original production method. So how does this Flt3 dendritic cell behave? We did some functional analysis. To check antigen uptake, we sorted those 3 dendritic cell population incubated with dextran DC at 37 degrees, with the ones incubated on ice as the negative control. The result showed that all of these 3 populations had the capability to uptake dextran. Well, in CDC1, 2 subsets were observed, indicating probably it is this population is heterogeneous. The antigen presentation was checked through mixed lymphocyte reaction. In this analysis, the sorted dendritic cells were co-cultured with pre-labeled PBMC at a ratio 1:5 for 5 days. Then stained CD3, CD4, CD8 and analyzed by flow cytometry. The result showed that there is almost no significant difference of these 3 populations in stimulating these CD4 and CD8 single positive T cells. While both CDC1 and CDC2 had a high capability to stimulate CD4, CD8 double positive T cells, with CDC1 showing the highest capability. In the following, we check the infection of this Flt3 DC by PRRS1 infection. Firstly, we infected the whole population of this Flt3 DC with a PRRS1 isolate 3267 at MOI 0.1. After 48 hours infection, 12% of the cells were observed as PRRS positive, with the titer in the supernatant reaching 10^6. Beyond our expectation, nearly all of these PRRS positive, nearly all of these infected cells are CD14 negative. The further characterization showed that these infected cells at the same time are also cadm1 negative, so in principle, these cells are not dendritic cells. Moreover, half of these infected cells are CD163 negative. This apparently contradicts with CD163 is the essential receptor for PRRS infection. To understand things more comprehensively, we sorted CDC2 and CD14 positive dendritic cells and infected them with the same protocol. Here is the infection of CDC2. After 48 hours infection, we can see that there is no positive cells detected by flow cytometry and no viable virus in the supernatant. So these CDC2 cells were not susceptible to PRRS1 infection. By contrast, this CD14 positive population can be infected with 6% of the cells labeled as PRRS positive and also viable virus in the supernatant. So why the infected cells of the whole population are CD14 negative, while the sorted cells give us a different picture? This might be answered by the change of CD14 expression. Here we can see that before infection, around 33% of the cells are CD14 positive, and after 48 hours culturing without virus, no big change, still 30%. That means only culturing didn't affect the expression of CD14. However, after 48 hours infection, this CD14 positive population largely decreased by more than 10%. So this indicates there might be a phenotypic change of this CD14 positive population during infection. So based on all of these experiments, and we can conclude, Flt3 ligand-based cultures mainly produce CDC2, a minor proportion of CDC1, and a CD14 positive population right now is difficult to classify. CDC2 were not susceptible to PRRS1, while CD14 positive populations were susceptible, but the infection might cause a phenotypic change of the CD14 population. Thanks for your attention.
So, questions for the dendritic study? Yeah.
I was curious, what multiplicity of infection did you use to infect? What is the MOI?
The MOI is 0.1 in all the infections. 0.1.
Why did you do that? Why don't you just high MOI?
If I increased the MOI, for example, to 1 to 10, it's almost impossible with my virus because it's difficult for me to get a high titer of this virus. If I increase to 1, a lot of dendritic cells, although they can't be infected, but because of maybe some cytokines created and they start to die, and the situation of these cells are really bad if I increase.
Right, so that's exactly the problem. If you are going such a low MOI, then you are depending on the replication of the virus to produce more virus to infect more cells so you can see them. So you go high MOI so you can actually see the thing. And within 8 hours or less, you should be able to see the infection. 48 hours is a long time, but you're using 0.01, so you require a lot. You're asking a lot for the system to give you the answer.
That's true, yes.
I'm wondering if you did a comparison with other bona fide dendritic cells that are actually isolated from tissues, because if I remember correctly, there have been isolations from trachea, spleen, and blood. And they have actually found that CDC2 dendritic cells express high amounts of the DEC205 receptors. And it's curious that in your research, that is actually lowered in that population. So I wonder how much they compare with actual bona fide cells.
That's really a good question, and it's also the things I'm thinking to do. But working with the in vitro production and at the same time to do the isolation from the pigs is really quite a big amount of work, and at this moment is not feasible in our lab. But it's a good idea that if we can compare these in vitro produced dendritic cells with the in vivo to see how different are they. I'm sure there will be some difference, like what you said, DEC205. And here the histogram I showed is only with one batch. And I found that really with different batches, especially with freshly isolated bone marrow hematopoietic cells to do the production, the proportion of DEC205 is much higher, is around 50%. So, but the histogram I showed here is really low because it's a very old bone marrow cells we stored in the liquid nitrogen.
Okay.
Yeah.
I can ask the last question. So you mentioned the infection of the cells can switch downregulate the CD14. So because the PRRS virus is a cytolytic, so do you think the lysis of the infected cells may play a role for the reduction of the CD14 expression?
Really, although I don't have the solid proof in hand, but it will be my future work to see that if there will be some phenotypic transition, not only for CD14. Actually, I found, for example, MHC2, CD163, CD11R3, and there is a transition from the double positive to single positive. And for example, DEC205, the transition is like from single positive to totally negative. So, and for the other, the other molecule, CD11R1 and cadm1, and there is a transition from double positive to double negative. And why, I don't know. This will be my future work.
Thank you very much.
Thank you.
Thank you.